Hey everyone, this is video number 1 for the lymphatic system. What I want to do in this video is to give an overview of the lymphatic system, general structure and function. And I prepared a short video to cover the first part of that. And then I'll cover the rest of that in the PowerPoint. So the first part shown here is basically overview of structure and function. And then the second part is going to be about lymphatic vessels, an introduction to the lymphatic types of lymphatic vessels. So let's take a look at this video. So I want to start out with a bit of an overview kind of introduction of the lymphatic system. First of all, what are the main functions of the lymphatic system? Two main functions. One is the lymphatic system is involved in returning fluids and proteins that have exited from the bloodstream into the tissues in the interstitial space. And so the lymphatic system involves a set of vessels, lymphatic vessels that are going to conduct that fluid back into the bloodstream, reclaiming it in a sense. That's number one. Second main function is somewhat related, but involves different structures, different organs, tissues. And that is the lymphatic system composes a bunch of different tissues and organs, which are sites where immune system cells, these are mostly the white blood cells or the leukocytes. Once they're made, they're going to take up residence and they're going to be exposed to stuff coming into the body, usually in a fluid. And so that main function is immune surveillance by the lymphatic system, organs and tissues housing these immune system cells. Okay? I'm going to explain this in the PowerPoint, but I wanted to elaborate on the first function a little bit more. How does the lymphatic system return fluids and proteins back to the bloodstream? So that involves going back to discussing blood circulation a little bit. And if you remember from the blood vessel chapter, this is the basic anatomy of a capillary bed. There's an arteriole leading into it. Here's the capillary bed, and there's a post-capillary venule draining it. In the capillary bed, there are two main things happening. There's fluid exiting, that's filtration, and that's how the tissues are able to take up substances from the bloodstream. So materials that exit are going to be used by the tissues. And on the other end of the capillary bed is the reverse. It's going to be taking, reclaiming most, not all of it, most of the fluid that exited here in filtration, about 90%, is going to be reclaimed by reabsorption on the venule end of the capillary bed. Okay? So that goes on all the time. However, there's a slight deficit here. 10% of what exited in filtration is not reabsorbed. So we can't have that constantly happening, loss of fluid from the bloodstream, even if it's a small percentage, 10%. That's going to accumulate over time, right? So there must be a mechanism to reclaim that fluid. And by the way, this fluid, 10%, totals up to about 3 liters per day. That's the total volume that's going to be exited from the bloodstream through filtration and not reclaimed directly back into the bloodstream. So this is where the lymphatic system comes into play. And there are a set of lymphatic vessels. There are lymphatic capillaries and larger vessels. Capillaries are going to take up this excess fluid. And then that fluid is going to travel through connected lymphatic vessels. They get larger in diameter. And then eventually, these lymphatic vessels are going to connect back into the bloodstream. And so the fluid in that way, you lose about 3 liters per day from your bloodstream, and you reclaim that same 3 liters through the lymphatic system back into the bloodstream. All right, so that's kind of an overview of the first function here, and then we'll pick up the second function when we start going through the PowerPoint. Okay, so number one there, overview of lymphatic system functions. And on the next slide here, that kind of breaks it down into the two main functions that I mentioned. Number one is the one that I just talked about in the video. Number two is the other main function. So lymphatic organs and tissues, the various structures in the body that are housing immune system cells. In some cases, they're also involved in the formation of those cells and the maturation, becoming functional. Okay? So that's the second main function. So let's break those down. And also, this mentions what these immune system cells are doing. They're performing immune surveillance, which means they're packed into these structures and they're exposed to stuff coming into the body. And they're basically evaluating materials coming into the body. That's immune surveillance. Is it good? Leave it alone. Is it a pathogen? Attack it. So surveillance, watching what comes in, and defense is mounting an immune attack on some kind of pathogen. All right, so this slide breaks down the anatomy, and on the left are the lymphatic vessels. I'm going to ignore that for now. I'll take that up in more detail later in this video and also in the second video. On the right are the lymphatic tissues and organs. So these are the structures that are either making immune cells, maturing those cells, or they're sites of residence of immune cells for surveillance and defense. You can break them down into two main types: primary lymphatic structures and secondary lymphatic structures. The primary structures are sites of formation or maturation of the leukocytes. Okay? So those include only two main places: the red bone marrow. That is the site for formation of all leukocytes, all immune system cells. The thymus is also included in this. The thymus is a site that's important for the maturation of a particular type of leukocyte called a T lymphocyte. We'll talk about that in more detail in the next chapter. Red bone marrow, site of formation of leukocytes. Thymus, site of maturation of one type. What do I mean by maturation? When immune system cells are first formed, they're not fully functional. So maturation is kind of a school for immune cells. They have to go through a process by which they become functional. And for one type of cells, the T cells, that happens in the thymus. Okay, the secondary lymphatic structures are shown on the right panel here. These are sites where the mature immune cells, now they're functional, they can do their job. And where do we want to put them when they're functional? We want to put them in sites in the body where they might be exposed to bad stuff coming into the body. So that's what these secondary lymphatic structures are. Sites that house mature immune system cells. And so they're therefore sites for immune surveillance, things coming into the body, and immune defense, battling pathogens, for example. So these would include the following structures: tonsils. Tonsils are in the oral cavity and also somewhat in the pharynx. Lymph nodes, and I mentioned I will mention lymph nodes in a little bit more detail in a second. And you find concentrations of lymph nodes, basically small pea-shaped structures in the neck region, cervical, armpit, axillary, and inguinal, groin region. Also, the spleen is an organ in the upper left abdominal quadrant that's considered a lymphatic organ. And finally, there's a set of structures called MALT, which stands for mucosa-associated lymphatic tissues. What's common to all of these is they have packets of immune system cells. The way they're organized differs in each of these structures. But in the MALT, basically is a group of immune system cells together, and they're embedded in a mucosal membrane. Remember that mucosal membranes line passageways in the body that open to the exterior. So this is a susceptible way for pathogens to get into the body. So that's a good place to have packets of immune system cells being exposed to stuff that could come into the body. So just to remind you, mucosal membranes are the lining of the digestive tract, of the respiratory tract, of the urinary tract, and of the reproductive tract. All of those are passageways that open to the exterior. Okay. So this slide is just a summary of the information that I mentioned on the previous slide. What are the primary versus secondary lymphatic structures? How are they different, and what are the tissues that are included in those? All right, let's switch a little bit now and talk about overview of the lymphatic vessels. So I talked about the function, how they work to reclaim fluid, but not exactly how that's done. So how that's done is through a set of vessels called the lymphatic vessels. And these vessels are tubes that are conveying fluid. They're similar to the blood-venous system in many ways. There are, the bottom of the slide here shows the names of the vessels, and similar to blood vessels, there are small diameter vessels that are numerous. Those are the lymphatic capillaries. The lymphatic capillaries are intermingled with the blood capillaries in any capillary bed. Okay? Lymphatic capillaries are where the fluid is going to enter into the lymphatic vessels. So they sort of suck this fluid in from the interstitial space. Okay? The lymphatic capillaries then are going to connect to a series of larger vessels by merging of the smaller vessel into the larger type. Many lymphatic capillaries are going to merge into a larger, what's called a lymphatic collecting vessel. Lymphatic collecting vessels are going to merge into one of 9 larger vessel called a lymphatic trunk. And then the 9 lymphatic trunks are going to merge into one of 2 lymphatic ducts. So in this hierarchy, vessels are going to get larger as we move down the list, but also fewer. Okay? So like I said, this is very similar to the blood-venous system. From a capillary bed, capillaries merge into a post-capillary venule, which merges into larger veins, which merges into the largest of veins, inferior or superior vena cava. So similar to how the lymphatic vessels are organized. Okay? This slide is showing that organization sort of diagrammatically. And here is the blood circulatory system. The heart is the pump through the arterial system to a blood capillary bed, then into the venous system back to the heart. So in the blood circulation is this continuous closed system. In green is shown the lymphatic vessels, and the smallest being the capillaries being intermingled with the blood capillaries, and that's shown better over here. So wherever you find a blood capillary bed, you're going to find a lot of lymphatic capillaries as well. Remember, there's going to be net exit of fluid from the blood vessels, and that fluid is going to be sucked up into these lymphatic capillaries. The lymphatic capillaries are going to merge to the larger lymphatic collecting vessels. They're shown here. They have all of these kind of bumpy structures on them. What those bumps are, these collecting vessels have internal valves. So there's another similarity to the blood-venous system. The lymphatic vessels are not under large pressure to move the fluid, which, by the way, is called lymph. Lymphatic vessels are conveying a fluid called lymph. The way this fluid moves forward through this pathway is partly through these valves preventing backflow. Another structure that's on the pathway of the collecting vessels is this. There can be one or there can be multiple. These are the lymph nodes. So these are one of the structures containing lymphatic immune system cells that are evaluating substances sucked up from the lymphatic tissue into the lymphatic capillaries and then conveyed into the lymphatic collecting vessels. Okay, the lymphatic collecting vessels are going to merge to form lymph trunks, which are going to merge to form one of two lymph ducts. Those are not really well illustrated on this slide. I have another slide which shows them better in a bit. But this does show that eventually the fluid sucked up into all these lymphatic vessels is going to be connecting into the bloodstream again. Okay. Now, one thing that's shown on the right here that's important to note is that the lymphatic vessels are a one-way system of transport. There's not a continuous closed system like in the blood. Lymph enters into the capillary and then it flows in a one-way pathway back to the bloodstream. So the capillaries are dead-end vessels. They're basically tubes that come to a dead end. And fluid is going to enter into these capillaries. They're extremely leaky vessels. They allow fluid to enter, and then it's going to flow away into larger vessels. This slide also shows a similar structure to the previous slide, but I want to emphasize what's on the right panel here. This is kind of a magnification of the end of a lymphatic capillary, and it shows that the capillary wall is very thin and very leaky. And the reason for that is it's composed of a single layer of squamous epithelial cells, shown here, and they form a dead end on one end of the vessel. And the neighboring endothelial cells are not very tightly connected to each other. They can flap wide open, actually. And so this ability, this is called mini valves, allows any substance in the vicinity to get sucked into this vessel. Okay? So that's a feature of a lymphatic capillary that you don't have in blood capillaries. Blood capillaries, the endothelial cells that are neighboring each other, are usually pretty tightly joined to each other, in most blood capillaries anyway. So this slide summarizes lymphatic capillaries, key features of their anatomy, and how they work. So like I mentioned before, they're similar to blood capillaries. They have a single layer of endothelial cells in their wall. However, lymphatic capillaries are extremely permeable, and they allow a large volume of fluid and even some particles into the capillary. The endothelial cells of lymphatic capillaries do not have an outer covering, a basement membrane, like blood capillaries do. Basically, the wall of a lymphatic capillary is just the endothelial cell, squamous epithelial cell, endothelial cell. And I mentioned that neighboring endothelial cells are not tightly connected. They have this flaps or mini valves that can flap wide open. Now, the mini valves act as one-way gates. And just to go back to this slide, they can flap open to let fluid in, but once the fluid moves in, they flap closed and don't allow it out. So this sucks fluid in, basically, by a pressure gradient. And then that pressure gradient will cause the flap to close, and then the fluid can only flow away from the capillary. So how does that work exactly? It's because of the third feature here. The mini flaps, the mini valves, the loose flaps on the ends of the endothelial cells have collagen fibers attached to the rim. And then these fibers extend out to other structures in the tissue. So let's see how these work in a situation when here is the flap, here are the fibers, they're called anchoring filaments. They're attached to some other structure, oops, sorry about that, some other structure in the tissue. Let's say you have a lot of fluid in this tissue. So there's swelling in the tissue for some reason. That creates a lot of fluid, and that fluid is going to put a lot of tension on these filaments. Tension is going to pull this mini valve flap open. When the flap opens, the fluid that's present in excess in the tissue is going to rush into the lymphatic capillary. Now, fluid has moved in. There's not as much fluid pressure out here. There's not going to be as much tension pulling on this filament. and that's going to allow these mini valve flaps to close. So fluid outside pulls on the flaps, opening them, allowing fluid to enter, which causes the flaps to close, and fluid can only move through the lumen of the lymphatic capillary. So that's basically how they work. What is lymph exactly? So lymph is the name of the fluid that's present in lymphatic vessels. Okay? It's an operational definition, meaning whatever's in the vessel is lymph. We can't really define very precisely what is present in lymph, because whatever's in the interstitial fluid is what's going to be in the lymph. Now, the interstitial fluid can vary in its contents from moment to moment in a given tissue, or from tissue to tissue in the same individual organism. So what you can say is lymph usually contains a lot of water, dissolved solutes. These can be small solutes like electrolytes, or even waste molecules or nutrient molecules, usually a small amount of protein as well. But not only dissolved solutes, the mini valve flaps are so large they can suck in really large materials such as whole cells or pathogens like bacterial cells or viruses. So even cellular debris, if you have some injury, that creates a lot of ruptured cells. Final slide here is just comparing a blood vessel capillary, let's say, to a lymphatic capillary. What are the contents? In a blood capillary, it's pretty well defined. When we talked about blood, there's a certain percentage of formed elements. And for all of the small solutes, certain percentage range for those, and for proteins as well. So you can pretty well define the contents of the bloodstream. Not true for the lymphatic vessels, for the lymph. Lymph is whatever gets into a lymphatic vessel in any given tissue. All right, so I'm going to end this video here. In the next video, I will take up the other lymphatic vessels.